Thank you very much for the introduction. I don't speak Korean, but I assume everything was very lovely. So thank you very much. Thank you everyone for joining this afternoon and coming to hear about the new updates that we have here at Bruker Spatial Biology, and it's very exciting. So I've been with the company for about 5.5 years. So we were NanoString before we were acquired by Bruker just last year. So I've been around and I've seen a lot of changes at the company and a lot of exciting innovations. And I'll share some of those with you today. So I like to show this slide a lot. It kind of shows, where we've come from in terms of spatial biology, in terms of the field and what we've accomplished as a company. So at AGBT, so this is a big genomics conference that's held, every year in the US. We've been attending for several years, but back in 2019, when we launched the GeoMx system, we were looking at 84 genes in space. At the time, that was 20 times more than anyone was doing at that particular time in 2019. We fast forward to 2020, we were up to 1,800 genes in space. Nature had dubbed spatial biology the method of the year. So it was a very exciting time as the field was starting to blossom. A year after 2020 on the GeoMx system, we were up to whole transcriptome detection. So the scaling is absolutely incredible. A year after that, we launched the CosMx SMI system. That was at 1,000 genes, which was higher plex than anyone was doing at the time. A year after that, we launched a 6,000 plex on the CosMx system, which is commercially available now. And we also launched 570 proteins being able to be detected on the GeoMx system. This year, what's really exciting, I'll show you some of these today, or give you a little bit of a vignette anyway, is that the CosMx system, we've actually reached whole transcriptome. So this is an absolutely monumental achievement. So over 18,000 genes are included in this panel that we're able to look at single cell subcellular resolution. And we can also incorporate that with up to 76 proteins, same slide multiomics, which is really, really exciting about the discoveries that we'll be able to make. And then also on the GeoMx, I'd like to announce there's a discovery panel as well that's going to be coupled with the GeoMx IO Proteome Atlas. So you're able to look at over 1,100 proteins in a single piece of tissue. So it's absolutely incredible. And that's where we're going. If you're interested in what you hear today and you'd like to learn more even after today, we will be running a galaxy tour. So that Joe Beecham, this is the inventor of our technology. He will be in Seoul, Korea, along with the application and R&D lead for our CellScape platform, which is our other proteomics platform. We will be in Seoul at the end of June, so I hope that you'll be able to attend and come and learn more about what we're doing. So this is our fleet of instruments that we have in the spatial realm anyway. So the GeoMx DSP that I mentioned before, we have the CosMx SMI, which I'll be focusing on mostly today, and also the CellScape platform. These are the two instruments I'll primarily be talking about. And then just a little bit of a sneak peek, we're also releasing something called the PaintScape platform. So this will be coming later in 2025. This is looking at spatial DNA, right? There's no other platform on the market like that, and there'll be more information coming about that later. But today I'll focus on the CosMx Whole Transcriptome Panel. So this is really an unprecedented assay that's been developed. But I just want to define for you what that actually means. So the Whole Transcriptome Panel covers about 19,000 RNA targets, and it provides full coverage across all protein encoding genes. And that's at single cell subcellular resolution in your tissues, right? The next closest thing to what we're able to do in this space is 5,000 plex, right? And what we can do with this, so another take home for you today, is that we can actually project complete pathway biology in space and map every cell-to-cell interaction at the individual transcript level. And we couple that with best-in-class sensitivity and genomic breadth within a single intact FFPE tissue section. And sensitivity, is defined by the number of transcripts that we can actually detect per cell. And genomic breadth is the number of unique genes. And if you're interested in learning more, you can scan the QR code here. So one of the most important things in any platform really is the chemistry and the approach that any company takes to being able to detect RNA or protein in the tissue. And what we've done from the very, very beginning, from the launch of our nCounter platform, which is a bulk RNA detection system that we have as well, is that we use hybridization chemistry, right? Very, very simple chemistry. So we look at about 35 to 50 nucleotides for an ISH probe that binds to an RNA molecule. And we use that to scale, right? So we use zero amplification, zero enzymology in all of our chemistries, right? It's a shared chemistry that we have, the CosMx system, for the GeoMx system, and for the encounter. It's the same chemistries that we've been using since 2011. We've been working on this for a very long time, and they were specifically designed to work in FFPE tissue. So we believe that that is the most important tissue that you can work with that's coming out of the clinic, and all of our assays were designed to work in FFPE tissue. Again, no enzymology, no amplification, to be able to perform direct molecule counting within the tissue. And it's that ability that gives us, or it's that chemistry that gives us the ability to scale. So in terms of the CosMx SMI system, it is the whole transcriptome assay is the first and only imaging platform to traverse subcellular spatial whole transcriptome biology on a single platform. As I said, the closest competitor that we have to us is 5,000 Plex. So the chemistry is very robust and scalable. So what we do with the CosMx system is we have an ISH probe, 35 to 50 nucleotides that bind to that mRNA molecule. And then to that ISH probe, we have a readout domain. And so what we do here is we have a number of reporters that will bind to that readout domain. And we go through a series of cyclic hybridization and imaging, right? And again, there's no amplification in this chemistry. All of these reporters are 20 nanometer. They fluoresce in 20 nanometers, and it's all scalable. So the way that we scale this is we just add some more real estate essentially to this readout domain, and we're able to add more reporters to that system, and we're able to scale that up to whole transcriptome. So what you'll hear for in situ platforms is that you need to worry about optical budget. And so that's true. And what that means is that when you're in an imaging platform, there is the really the physical limitation of saturating your detectors, right? And so that's why a lot of platforms have the difficulty in scaling to whole transcriptome. We don't have that problem because, again, we don't amplify that signal, and we're able to control that with our reporter probes. And that's allowed us to plex at a very unprecedented level. So again, our assays are designed to work in FFPE. That's the way that we worked with them. As I mentioned, we're looking at the protein coding transcriptome with this particular offering in humans, for human samples, excuse me, and we also use accurate cell segmentation. And that's also an important component, right? So what we find is, one of the most, the important component for spatial imaging is accurate cell segmentation, right? That's one of the leading causes of error. So being able to look at these different things and different approaches to spatial transcriptomics is important. So on the left-hand side here, we have an imager. So this is looking at, hybridization chemistries, looking at direct molecule counting within a tissue. We have a profiler here in the middle. So this is the GeoMx system where we're looking at, multiples or an aggregation of particular cells. Now, what's unique about, the SMI and the GeoMx system, what we have here is that we've designed these systems to actually outline the contours of actual biology within the tissue itself. So we can outline individual cells or single cells, and we can accurately reflect what those look like within our systems. Now, if you can compare that to an array-based type of system that you use for spatial transcriptomics, what you can see here is if you have a gridded approach with these arrays, right? So here we've got a 3x2 array. If you can look inside of this array, so this is a binning strategy that you would use to capture the transcripts out of that individual cell. You can see that it's not actually a single cell, right? So there's this cell here. There's about 4 or 5 cells that are actually captured within this little bin. And so what that does is it misallocates transcripts from different cells into one particular cell. So what that does is it gives you transcripts that don't actually belong there, right? And that's a limitation that you can't get over computationally or through other means either. And so that's a limitation of an array-based approach. Okay. All right. So what we've done essentially, since the beginning, is that we've always used a multimodal type of approach. So in our spatial molecular imager, we stain the tissue with protein markers to accurately define the nucleus, cytoplasmic components, and also markers to outline the cell boundary as well. So we are on our 5th generation of this AI machine learning model, and we train all of our models in-house with specific tissues. So we manually curate those to get the best segmentation possible out on the market. And we've shown that in numerous occasions. If you look over on the right-hand side, this is an example in a mouse brain. And what you can see, sorry, this is human brain, apologies. But you can see with our segmentation approach, we can actually outline, different projections and unique biology that comes out of every individual cell. So you can't do that with a nuclear expansion model, and you certainly can't do that with a gridded array. So there are 4 main points that I want to emphasize for this talk. So in terms of the design of our platform, we've always wanted to plex to a high scale, right, in both protein and RNA. And that's because we don't really understand how tissues work, and we're trying to discover as much as we possibly can out of there, grabbing as much information content as we possibly can. We are focused on FFPE, as I mentioned before, because that's the most important clinical material. Segmentation, as I just mentioned, is extremely important. And then also, when you're generating these large data sets, especially whole transcriptome imaging data sets, you need to have the analytics and storage capacity to be able to scale that chemistry and be able to understand what's going on. So in terms of CosMx, it has the highest sensitivity. That's the number of transcripts detected per cell is the highest in any platform. We have the highest Plex, and it's the highest Plex multiomics technology that's ever been accomplished. It's actually truly phenomenal what we're doing here. I'm very proud to be a part of that. Our atomic system, this is our information portal that we have where you can interact with your CosMx data. But it's also the continuum that's extremely important. It's coupled with the CosMx system to be able to decode all of those transcript data as it comes off the instrument in real time. And right now in atomics, we have over 1.8 billion cells, over 6 billion transcripts, and 2,000 terabytes of data that we've been able to collect across the entire world with customers that are using CosMx. And finally, 4, we're breaking the 4-digit protein plex barrier for spatial multiomics at scale. And that's on the GeoMx system, where we're able to look at over 1,100 plex protein in addition to whole transcriptome on the same tissue. If you're interested in learning more in terms of doing a deep dive on the whole transcriptome assay, you can look at our preprint here. So this has been released. I was actually in the car with Joe Beecham when he posted this on LinkedIn when it came out, when the preprint was up online. But here we looked at over 6 different FFPE samples across different tissue types, so skin, hippocampus, breast, kidney, colon, and pancreas. The number of transcripts, we're actually capturing about 1,500 transcripts per cell and over 900 unique genes per cell. We'll have publicly available data sets for multiple tissue types that will be available soon. All right. So I mentioned earlier that one of the things that we can look at with the whole transcriptome is kind of redefining the way that we start to look at tissue biology. So, when you're thinking about in situ experiments, a lot of people want to just look at cell typing, but what we can do with whole transcriptome level is we can actually project an entire RNA pathway onto a particular tissue. So, what can you do with this level of Plex at subcellular resolution? So it turns out that you can project the entire tissue biology at the RNA pathway level for completely new functional understanding of your samples. Here we show complete coverage of 4 conical pathways projected onto a colon cancer sample, which you can see on the left. It's kind of covered up there. But note that some of these pathways contain a number or an aggregate of genes. So if we're looking at EGFR here, so this is an important pathway in cancer, as you know. So what we're doing here within the heat map, you can see these pockets, hot pockets of EGRF expression that you can see within that tissue. And this is the aggregate of all of the 100 genes within the EGFR pathway that we've projected onto the tissue itself. We can do that for VEGF, we can do it for T-rail, we can do it for TGF-beta, or any particular pathway that you're interested in. And we can actually look functionally at what's happening within the tissue. Right? This is hypothesis-free, just looking at what's actually happening in the tissue based on what we're measuring. All right, so here's an outline of that as well. So, with that coverage, we're now able to generate molecular fingerprints for each tissue sample. So, for instance, the upregulation of growth factor pathways, the EGFR and the VEGF there, coupled with the downregulation of the apoptosis-inducing T-rail pathway, suggests that the tumor region in this sample, which you can see outlined there on the left, exhibits characteristics of an aggressive, progressive, evasive colorectal carcinoma. So this is the kind of information that you can get, and it's really exciting. All of this as well, I should mention, you can perform within NtiOmics, and we have scripts and guidance for you to download this data for your own use through third-party tools. So the other thing I think that's interesting about the whole transcriptome assay is that shifting the paradigm of understanding how we perform spatial analysis using this technology, right? So usually the first port of call with most researchers that are working with sort of your mid or low plex kind of in situ assays, so 100 plex, 500 plex, is that you look at cell typing, right? So where are my cells? How are they situated within the tissue itself? And then you start to ask questions about that and you infer that biology. So what is actually happening within that tissue, based on that low plex data that you have. But with the whole transcriptome data, you don't have to infer anything. We can actually directly measure what's happening within the tissue itself. So on the right-hand side here, this is an adenocarcinoma sample, I believe. But what we're doing here, before we even perform any cell typing, which we can do, and what you can see here on the left, is that you can look at actually what's happening within the tumor itself. So in this particular example, you can see the primary tumor is over here on the right, or excuse me, on the left. And on the right-hand side, you can see an invasive margin with some of the immune cell stroma. And so what you can see in this sort of tan covered area here is a number of proliferation and invasion reactome pathways as the tumor starts to move through the tissue in this normal tissue. Behind that, what you can see within this invasive region in the light blue domain are all of these pathways and characteristics of a stress response to nutrient deprivation as the tumor cells start to grow and move through that tissue or the tissue itself and start to destroy it. So these are things that we can look at just projecting the whole reactome pathway onto the tissue itself. So not only that, but we can start to look at individual cell types and start to get a deeper understanding of what's happened. So in this particular example here on the left, you can see in those black dots here, these are activated CD8 T cells are concentrated in two spatial domains. So in the stroma bordering the tumor and the stroma inside the tumor. So we can ask across every gene expressed by CD8 T cells, how do CD8 T cells change as they enter into the tumor? So each differential expression of each particular gene, absent from commercial gene panels, represents a new understanding of this biology. And so what we can see here is as these, excuse me, so the T cells that are enriched in the tumor interior, you can see that a lot of these different markers will come up, these different pathways. And of course, you can see T cell activation and survival, which makes some sense as the T cells start to move within the tumor. And then we also see that there are genes that are depleted within the tumor interior. So for instance, you have GMAP2. So this is for T cell survival. This particular marker as well is not included in any other high-plex spatial panel. And we can also look at GZMK, right, for cytotoxic killing as it's starting to battle the tumor itself. But essentially what you're doing here is we're measuring things that you wouldn't even know were there if you didn't have that level of high plex to be able to understand what was actually happening. So here on the left-hand side too, you can see this is an example of breast cancer. Within the region, we have two areas of tumor. We have in the proximal and distal areas of the tumor. Looking at the T-rail and JAK-STAT pathways, you can see that there's a variation in expression of these different pathways between the tumor itself. And if we look at JAK-STAT, you can see that there's a hotbed region for JAK-STAT pathways in this lower portion of the tumor, and in the top part, in the proximal area of the tumor, you can see that it's cold. So we can start to understand what functional things are actually occurring within the tissue itself. So we have over 2,750 of these reactome pathways that are included within the panel to really explore your tissue and start to understand what's going on. Right, so the CosMx Whole Transcriptome assay, it's really unprecedented in terms of what it's able to accomplish. So the three takeaways that you should have here is that you get a direct and complete projection of pathway biology in space. So this is a paradigm shift from inferencing biology based on just cell types alone in low plex panels. You're getting best-in-class sensitivity and genomic breadth designed specifically for FFPE samples. So you get robust gene discovery, comprehensive phenotyping, spatial domains, and an understanding of your tissue. And you're really able to create a molecular fingerprint from every patient sample about what's actually happening. So this is extremely important for understanding, variations in, disease or cancer subtypes, and understanding how specific patients are responding to a particular treatment. So I will walk you through a couple of comparative studies. So, our probably fiercest competitor, I guess I would say, is 10x Genomics. But this is a comparative study that has come out of Parambir Dulai's group at Northwestern University. He's a clinician and scientist. So in this particular study, he did a comparative study between the CosMx and Xenium platforms. And this is the most robust comparative study that's been done. So he looked at 16 individual blocks across 11 different patients in ulcerative colitis and Crohn's disease. So what he did here was he standardized the collection of these tissues, put them onto slides, and then ran them on the CosMx and the Xenium system at the same time, which is extremely important. So the Xenium run was done at the Northwestern Core facility, and the CosMx was run in his own lab. All right. So after he did that, he did an analysis of the relationship between the data, and this is what he found. So on the left-hand side, we're looking at the CosMx 1K gene panel and the Xenium 377 gene panel. So on the left-hand side, you can see in terms of both panels in their entirety, in terms of the number of mean transcripts per cell, were 5 times more, and this is including every single target within our panel. In terms of the mean unique genes that are identified within the tissue, over 6 times more. Now, if we look at the overlapping genes and transcripts within each of the assays, they're roughly commensurate, right? This is true. But as we start to scale, right, so he ran a 1K 377 panel. He also ran the CosMx 6K and the Xenium 5K. So in terms of looking at the entire panel as a whole, 5 times more transcripts, 4 times more genes with the 6K panel when you look at the panel as a whole. In terms of overlapping genes, you can see that it's 3 times more and 2.5 times more roughly for unique genes in comparison to the 5K and 6K. And I don't have the data for you now, but as we scale up to whole transcriptome, this also starts to change as well. And again, CosMx, we're identifying more unique genes, again, up to 1,500 unique transcripts per cell and over 900 unique genes per cell as well. So we're able to scale. And what that means is that we're actually able to compete or look at metrics that are very similar to what you would get with single cell RNA-seq. Right. So in terms of turnaround time, we've made a lot of market improvements with the CosMx system and with AtoMx. So from our commercial launch, really in January of 2023, the CosMx is 2 times faster since we launched the system all those years ago. So for a 1K panel at 1 centimeter squared, this used to be around 8 days or so, 7.5 to 8 days. We're now down to 5 to be able to run 2 slides at 1 centimeter squared, which is roughly industry standard. Our 6K panel, it's a 1 centimeter squared, is roughly 8 days. And our whole transcriptome assay is roughly 11 days for 2 slides at 1 centimeter squared. And again, you're getting a 19 times increase in the gene content with only roughly 2 times increased in the turnaround time based on what we've seen. All right. So this is exciting. This was just an announcement that we made today. AACR is going on in Chicago in the US today. And so this is a press release that we put out today. So this is the launch of what's called our CosMx 2.0 upgrade with our AtoMx platform. So the advancements that are made is that the CosMx SMI is 2 times faster. And so we've improved our decoding algorithms and performance of the software. So for any of you that own a CosMx instrument, this software upgrade is free. And also what CosMx 2.0 enables is the ability to look at same slide multiomics with the CosMx SMI, right? So that's whole transcriptome information with 76 proteins, all on the same tissue itself. And we're seeing a 2 times increase in the RNA detection efficiency. So improved metrics for sensitivity and specificity on transcripts detection per cell. So this is just some examples and just a brief vignette into why we're looking at same slide multiomics and why it's important. So this is looking at the 76 plex protein here on the left within a tumor sample. So here you can see we've got a tumor in the bottom right-hand corner, and we have a tumor up in the top left-hand corner. In the middle is the immune-rich cell stroma. So this is protein information. On the right-hand side is our whole transcriptome. And of course, we know that there's an abundance of proteins, right? A lot of proteins within the tissue, but it doesn't give you as large of coverage because we can't get to a full proteomic coverage. But transcriptomic information, of course, we can get up to whole transcriptome. It gives you a lot of useful information, but it can be sparse, right? And so if we look in this particular area, we see some areas of the tissue where we don't see any kind of, really too much transcript expression activity. But as we start to layer some things through and we work through this tissue, if we look at fibronectin, for example, within this tissue, in this protein, right, we can see that there's a bright layer of fibronectin that's actually being expressed between the tumor and the immune cell stroma. So this is creating a functional, physical barrier between the tumor and the immune cell stroma when we start to look at the protein data itself, right? Looking at smooth muscle actin, you can see that we have quite a significant expression of smooth muscle, which is starting to form around the tumor area in the lower right-hand level. And then we have transcripts that match up with that, so that's important. And what you can see here is that across the fibronectin barrier and also the smooth actin muscle area, when we look at B7H3, which is a checkpoint inhibitor, we can see that those are expressed along the tumor invasive front between the immune cell stroma. We can also see expression of IL-18 within the tumor itself, and then that's matching up, quite nicely with the transcriptomic data that we're seeing. We can also look at STING as well. So what this is able to do is give us a real comprehensive understanding of the biology that's at play within the tumor microenvironment, looking at both RNA and protein in the same area. So what we're looking at here as well is the ability of looking at whole transcriptome with the 75 plex proteins. This also includes phosphospecific proteins within our panel, and the ability to register H&E images with that as well. So we have some algorithms that we're working, and that's coding. Sorry, just excuse me one. Hi, apologies, everyone. I had an electrician show up, so one of the pitfalls of working from home. All right, so here, basically what we're able to do is co-register both RNA and protein and the H&E image together and create these comprehensive images as well. So we're working with a group from Fabian Thies's team to make sure that we can scale this and use this within AtoMix itself, but also provide it for use in open source tools. Okay. So, the interesting thing, considering single cell RNA-seq, we know that the endpoint of that assay is the UMAP, right? So we're looking at the UMAP here on the left. There's no spatial information here, but we're looking for differences in cell type populations based on expression label data that you pull from single cell RNA-seq. But what the CosMx now does at whole transcriptome is it actually gives you the ability to understand where these cells are actually located in space, right? You get spatial information. You don't lose any of the cells from disassociation. Everything is captured within this particular assay. So we've looked at some comparisons between CosMx whole transcriptome and chromium assays as well. And the sensitivity in genomic breadth is really comparable to what you can see out of a chromium. So in this example here, we have a chromium run that we've done. And the starting material is 2 by 25 micron sections. And then for the CosMx run, we've looked at one single 5 micron section. And if we compare the metrics there, the total number of genes detected are comparable. The input sample size, so the output for a chromium sample from these 2 by 25 micron sections, which could be precious samples, is roughly 10,000 cells. We're looking at 400 FOVs. So these are areas of the tissue that we define to look at using the CosMx system. The output number of cells here from a chromium system, 6,000. For the CosMx system, nearly 500,000. Right, so the median counts roughly the same, median features roughly the same as well. Genes above background, more for us. And then in terms of dynamic range, they're roughly the same. And the coverage of the tissue composition obviously is biased because you're disassociating with the chromium, but with the CosMx system, you maintain spatial integrity and are getting more information directly out of the tissue. Right, so you don't have to do any follow-up assays after single cell RNA-seq to move to spatial. You can do everything on the CosMx system itself. So in terms of the total transcripts detected per cell and the unique genes detected per cell, you can also see across a number of different cell types that they are commensurate when using both panels. In this example here, we're looking at pancreas tissue section. And what you see when we start to compare the metrics, again, the output number of cells, 8,000 roughly for the Chromium system, 400,000 for the CosMx system. Median counts per cell and median features are roughly the same. But what is important here is we start to look at specific cell populations. So as I mentioned, when you dissociate the tissue with single cell RNA-seq, we know that certain cell populations won't survive. So what we found with the Chromium versus the CosMx system, when we're looking at the pancreas here, is that alpha cells, beta cells, delta cells, and gamma cells, you're able to get counts of those on both platforms. But when you're looking at epsilon cells, which comprise less than 0.05% of the tissue itself, those are lost in single cell RNA-seq, but you can pick them up with the CosMx system, right? So maintaining that spatial data, maintaining that important information to your biology is also extremely important. So what we have here as well is a concordance of CosMx with Chromium data, and you can look at cell composition. It obviously changes when you perform tissue dissociation. So if we look at some of the percentage of the cells that make up that particular tissue sample, if we look at stromal cells, lymphocytes, macrophages, and tumors, they're roughly commensurate between both platforms. But if you look at smooth muscle, goblet, and epithelial cells with the Chromium system, you'll see, there's about a 10% or even, more difference between those cells. So you really pick them up, and you're actually missing out on some of the biology that's extremely important. We do have a poster from AGBT this year that will be coming out that's highlighting this study, so you'll be able to look at it. There is a group out of the University of Queensland. This is from Arutha Singhe's lab. So this is actually here in Brisbane, where I'm located. So this is a study that was done on CosMx Whole Transcriptome. They looked at 5 different TMAs with patient samples and over 600 patient samples. And what they found with this assay, so this is looking at a variety of different things in lung cancer. But what they found when they compared this with single cell RNA-seq data is that the genes detected across that comparison between the CosMx and single cell RNA-seq commensurate. Right. And again, with CosMx Whole Transcriptome, you're able to get that spatial data and all of the pathway projections and information that I mentioned to you earlier. This is now being written up and should be in press within the next couple of months, I believe. In terms of the AtoMx platform, so this is the continuum of informatics continuum of looking at spatial informatics data and creating this comprehensive ecosystem where we can actually decode the CosMx data within the cloud, and then we're able to move that either through analysis in atomic space. So you can use that depending on your level of comfort with bioinformatics analysis. We have a number of tools you can use within our platform. You can also customize it as well. But of course, for those of you that are bioinformatically savvy, you can download your data, and our data is completely portable and can be used with a number of open source tools. So R, Python, SquidPy, whatever it may be. We're also developing new spatial algorithms for single cell, and I'll show you those in a few minutes. But we're continually trying to develop this. But this is really a tool, essentially, it's able to decode that high dimensional transcriptomic data and proteomic data in the cloud. It's extremely important, that compute power. But then it serves as a repository for different studies as well. So when we're looking at these high plex and high dimensional assays, it's also extremely important to understand that they require different computational approaches that are fit for purpose. So we have, again, hybridization, in situ hybridization chemistry that's not amplified at very high plex data. So it doesn't necessarily fit in with traditional approaches that you would see with single cell RNA-seq or other amplified approaches in spatial biology. So we've actually created tools that can help guide you in that endeavor. So I'll make 4 points here. So the first one is looking at cell typing. So we don't base our cell typing and CosMx data on your sort of canonical gene marker kind of assay. So just looking at a few different genes. What we do is we leverage the entire gene panel itself, right? So that's 1,000 plex, 1K, or the whole transcriptome itself to create a unique transcriptomic fingerprint for each individual cell type. So we use the RNA data, and then we also use protein data as well to feed into that algorithm that's called in situ type. So this is in preprint, and then we also have the script is available too on Scratchspace, so I can share that with you if you're interested in reading more about it. You can leverage this to perform better cell typing using CosMx data. Another important aspect of defining cell types is actually creating cell type matrices to leverage within the tissue itself. So what we've done is we've provided reference guides for you. So as you go through your experiments, and we also have some information. So each disease type and each tissue type has a unique reference profile. And what we've done is trying to help you create that traditionally based off of reference guides from single cell RNA-seq that we've used to build those tools out. Following on from that, what we've also done is try and focus on creating CosMx-derived cell profile matrices for incorporation of direct hybridization. So remember, we don't have any sort of amplification strategies, and that can be a confounding factor when we're performing or creating an accurate cell reference profile. So we've encouraged our customers to build their own through their experiments with CosMx SMI, and we've also provided a repository for you to download these and use them on your own. And then finally, looking at subsets of genes through the hierarchy that we've created. So essentially within this, we don't use all of our genes within the whole transcriptome panel to perform cell typing. So a lot of those are used for pathway analysis, as I mentioned earlier. But we are able to curate and use specific subsets of genes to identify unique subsets of different types. So, CD4, CD8 cells, for example. And this is a tree-based clustering approach that we used. And again, this is all available through Scratchbase and can be used to help guide you. I'm just going to talk about the genomic system just briefly here. So this was the first system that we launched, as I mentioned. Just some updates here. So some of you may know we launched, I think it's about 1.5 years ago, the Human IO Proteome Atlas. So this is 570 plus proteins within tissues that you can identify within specific regions of interest, right? And there's over 64 or 68, I believe, phosphospecific proteins, which are in this panel as well. And you can spike in up to 60 targets if your specific targets are not located within this. And we've tested this across a variety of different tissues. Of course, this has all been validated, and we have a number of customers that are running these assays right now. As I mentioned, we have a number of phosphospecific proteins within here. So we validated all of this for you. So in this particular example here, this is the entire panel itself, which is phospho-treated. We're not looking at the phosphospecific genes in this particular plot. So you can see that there's no drop-off in performance. When we do add a phosphatase to the tissue, you can see the phosphospecific genes are actually dropping off, so the performance is maintained. And I will also state that we have same slide multiomics with this panel as well, so you can perform whole transcriptome analysis, 18,000 plex RNAs plus these 570 proteins on the same slide. And we have customers in Asia that are running this right now, which is very exciting. So the reason that this is important for same slide multiomics, obviously protein and RNA, they don't always correlate, particularly when we're looking at post-translational kind of modifications. So in this particular example, we're looking at 8-week human embryo data. This is taken from a lab in Amsterdam that we're collaborating with. So for this particular example, you can see that there's a nice concordance between the protein and RNA expression. But on the right-hand side, you can see that we don't really see that correlation at all between the RNA and the protein. So it's important to have both layers of information. And of course, if you're looking at phospho-specific targets, or if you're interested in those, we can't look at those using RNA alone. So one thing I'll just show you really quickly what we launched as well. We have some information on this at AACR is the launch of our our new 1250 plus protein panel. So this is our discovery protein panel. And this is, looking at over 99% of the proteins expressed in over 40 different tissues that we've looked at. So you can see those that are outlined here on the left. And we have over 120 plus protein specifics, or proteins that are specific to post-translational modifications within this panel as well. And this will be commercially available at the middle of the year. Okay. So the last portion of my talk, I'm going to talk about our CellScape platform. So this is our new proteomics platform that was launched. This was originally under the Canopy banner. but now they're under Bruker Spatial Biology. So with the CellScape platform, it's the only protein platform where you have quantitative performance, and we're able to accomplish that via high resolution and high dynamic range. I'll show you what that looks like. It's a very straightforward and reliable platform, so it's very robust in the way that you're able to develop assays and run assays on the platform. You can look at any marker, so it is an open platform, so you can use your own clones. And we do have some off-the-shelf panels as well that can help you with your assay development. You're able to re-examine studies as well in slides and store them and come back to them. And we have the largest imaging area on the market as it stands. So this is the typical workflow. You'll have just a standard slide that you get a positively charged slide you get out of the histology lab. You put a flow cell over the top of it. And then you go through a series of staining. So stain markers, you can perform up to 4 at one time. So you stain the tissue itself, you image, you then quench the signal, and then that process repeats itself. And then we have an on-instrument data analysis suite called PowerOmics that we use to look at the data, which was just released. So the quantitative performance of CellScape really sets it apart in the field of spatial proteomics. So this quantitative performance is the product of 2 technical attributes, which is resolution and dynamic range. So we can capture images with 182 nanometers per pixel of digital resolution. And this is combined with our proprietary HDR image acquisition method, which is described in more detail a little bit later. And this generates images with 8 logs of dynamic range. So this is important because it allows users to detect and quantify the full range of protein expression in biology, enabling users to not just detect the presence or absence of the protein target that you have of interest, but truly quantify protein expression across the full continuum, looking at both high and low expressors. All right, so we're able to generate 8 logs of HDR images by taking the same image multiple times using different exposure times. All right. So the long exposure times are used to quantify dim signals, but in the long exposure images, which you can see on the bottom right, those are bright signals, those are saturated. So you see the red pixels in the images on the left on half of the slide. So when the exposure times are reduced, the bright signals are quantified within the linear detection range, but the dim signals are below the noise floor or undetectable. So there is no one single exposure that is able to quantify both these high and low intensity pixels. So multiple exposures are necessary. The multiple exposures are then overlaid onto one composite image, which I'll show you here just on the right. And you can see with our broader dynamic range overall, we're able to pick up these high expressing and low expressing targets and be able to quantify them. So we have the best in class resolution as well. So you can see on the left and in the middle, these are 2 of our competitors. We're able to get down to 182 nanometers per pixel. So you can see on the left-hand side, these are kind of pixelated and a bit blocky in terms of being able to analyze each of these individual cell populations. We're looking at FOXP3 in pink and CD45 in white. And you can see with our platform here that you can actually see punctate dots for the FOXP3, and you can see individual projections of CD45 and the actual contours of the cell itself. So this is one of the main benefits, is the resolution that we have on our platform. As with all things, chemistry is extremely important. So some of our competitors will use indirect immunofluorescence. So this is secondary labeling of their primary antibodies. So one of the pitfalls of this is that you have cross-reactivity at high plex. And to remove these antibodies, it's also requires very harsh antibody stripping, which limits the plexing capabilities of that particular platform. Right, other platforms will use oligotagged antibodies that are conjugated to a fluorescent label. So this can restrict antibody selection to these oligo-conjugated variants, and custom conjugation may impact the affinity or binding specificity of a particular antibody that you're choosing. This approach also can add significant time to assay development and validation. So typically with customers that are using this approach, that process may take anywhere from 6 months to over a year to be able to develop that assay. So we use direct immunofluorescence. So these primaries are directly bound to the epitope on the tissue itself. This is a highly specific approach. There's no extra steps involved, so there's less time to get an assay up and running. And we also use a non-destructive photo bleaching method that can remove this signal, and it's very, very gentle on the tissues. And I'll talk to you more about that in a little bit. So we also have the largest imaging available on the market as well. So one of our competitors, this is one of them, it's looking at 13 by 13 millimeters. So this is kind of one of the limitations of one of our competitors. For us, we have a 44 millimeter by 16 millimeter scanning area. Right? So we're able to capture a number of tissues on there. So TMAs, multiple tissues, you can put onto our system, and the system can accommodate up to 4 slides at once. We also have the ability to expand the assays and test your hypotheses in real time. So essentially what you can go through, you can probe your assay for, let's say, 10 to 12 markers. You can then store that for days. Oh, excuse me. You can then store those slides within storage buffer in the fridge for days or even months later. And then say, your PI or a journal comes back to you and says, hey, it'd be interesting if you looked at these additional markers. You can then take that slide back out of storage. You can reprobe that same slide, and then you can get the answers that you need. Right? So you're able to optimize your assays, screen them for suitability, for subsequent assays, and also make data-driven decisions. Right. So we use photo bleaching in order to remove the fluorescent signal on our antibodies, and that's very, very gentle on the tissue. Here you can see an example. We've got CD3 markers. After 20 rounds of photo bleaching, tissue integrity is maintained. This is looking at a competitor that's using indirect or secondary antibody labeling. After 20 rounds of stripping, they have very harsh conditions. You can see that it starts to eat away at the tissue. And you're not able to do any subsequent assays and may not even be able to complete the assay that you're running in time. So just a few months ago, we launched a new buffering system, and it's called EPIC-IF technology. So this is a very gentle yet powerful signal removal capability that we have to remove fluorophores from antibodies on our system itself. And I'll show you what that looks like. So the EPIC-IF solution, it allows filtered photo bleaching, which is what we use, and the optical background subtraction removes the signal between cycles. So essentially, when we add EPIC-IF solution to that, when we're erasing the signal, it's something that happens within 5 to 15 seconds, rather than a long amount of time. In addition to that, in terms of how we perform our photo bleaching, we use filtered photo bleaching. So other platforms will use UV light or IR, which can damage the tissue. We filter IR and UV out of that, and we use visible light to remove our signals, which makes it also gentle on the tissue and allows you to run subsequent assays on the tissue. So that could be, you could look at doing a transcriptomic run, in situ transcriptomic run, and run that on the CellScape system, and the tissue will be maintained. So in this example here, I think, excuse me, I'll just need to pull this off. Here's a real-time example of using EPIC-IF. So on the top left-hand side here, you can see EPIC-IF. This is an anti-CD3 AF488 marker. When we don't use EPIC-IF, you can see in the top-hand panel here, the performance of the assay, or excuse me, the intensity of the signal. When we add EPIC-IF to that, you can see that the signal is erased in real time. That was taken down the microscope. On the bottom side too, we have a Cytox orange. It's a DNA stain as well. When we add an EPIC-IF to that, we start to remove signal, but just in a matter of seconds, that signal is removed. Right? So what this does is it allows us to cycle faster and increases the turnaround time on the platform itself. And it also opens up the ability to use a number of organic dyes on our platform as well, which gives you more flexibility in the antibodies that you can choose. Just time. So yeah, there's over 6,000 antibodies for FFPE tissues that are readily available for direct IF detection. So this can streamline your development. So this opens up the amount of things that you can do. One of the last points that I'll talk about in terms of turnaround time with our system is when you're thinking about running an assay on a system, it's not only the time on the system, but how long does it actually take for you to optimize and develop an assay? So using direct immunofluorescence, we use primary antibodies. Those are highly specific. There's no extra steps that are involved. You get a faster time to result. And we use non-destructive photo bleaching, which means you can plex at a very high level. So here's a couple of examples of studies that we've run in-house. So this is a colon cancer sample, 55 plex assay. 15 of the clones that we used, we did not know, right? We had not used them. The time that we had to result was less than 2 weeks, and our antibody success rate was roughly 90%, right? So that's the optimization, right? Instead of months, we were able to accomplish this feat in just a matter of weeks. On the bottom level, we've got a human brain sample that ran a 30-plex assay. 22 of those clones were unknown. The time to result was less than 1 month, and our antibody success rate on the platform was 85%. So we do have some off-the-shelf panels as well. So these are called VistaPlex panels. So at the moment, we have an immune profiling panel, tissue architecture, and immune cell subtyping. I can send you this information, or you can look on our website for these particular targets. So you can use these, and then you can also use them in combination with your own antibodies, or you can use your own antibodies on their own, right? So you can use primary monoclonal antibodies, and or if you have secondary labeling that you want to do, you can also use that on the system as well. Okay, this is my last slide. So we have a lot of flexibility on the platform as well. So you can look at proteins and RNA-ish technology. So this could be ACD bio, using RNA scope in addition to what we're doing. You can run proximity ligation assays. So, those that are provided by DaVinci or Hawk Bio, you can run those on our platform. And then you can also run, use different approaches using secondary antibody labeling should you choose to. So there's a lot of flexibility with the platform, and it allows you to, essentially do what you want. A lot more flexibility than you would get with the CosMx or the GeoMx system. And that's really one of the powers of the system. So that's my last slide. Thank you very much for attending. And if you have any questions, feel free to sing out or put them in the chat. Thank you very much. 감사합니다.